Hello, and thank you for watching today's webinar From Concept to Market, Overcoming the Challenges of Manufacturing and Clinical Trials, presented by Altasciences. Our speakers today are Kevin Kirkcaldy, Pharmacy Lead from our Montreal clinic, and myself, Angel Reyes. So for our agenda today, we're going to talk briefly about Altasciences' evolution and the services that we offer, and those that we've added over the last several years. Then we will get into the heart of the webinar, which is our Pharmacy on Demand services, Pharmacy GCP services, and our GMP manufacturing services. We will discuss individual case studies that show how the teams can handle complex situations and complex drug product manipulation within their facilities and their own procedures. What is most important is how we can share those activities across our organization and really provide creative solutions for our clients who are often up against some pretty important challenges. So a little bit about Altasciences. We are a mid-size, full-service CRO that has been around for over 30 years. We spend most of our time in early stage development where we can take you from lead candidate through proof of concept. With our CDMO team in place, we can also take you beyond that through commercialization with our manufacturing expertise in this area. Our leadership team is set up a little unconventionally compared to the CRO setup that you would normally see. We have deliberately done this to ensure that all of our lines of business are reporting to two executives who work extremely close together. This ensures that all of our services are very well knitted together in terms of how we provide the services and how we communicate the client's needs across those lines of service. We are located in North America with physical sites in the United States and Canada. Our 3 clinics are all dedicated to early phase clinical research and of course have pharmacy operations embedded within the facilities. The CDMO will be discussing their facility and capabilities later in the presentation. We are just over 1,000 employees, and you will see on the next slide that we were founded in 1992. Employees have come from within pharma, biotech, academia, and competing CROs over the years and bring different experiences to our team here at Altasciences. A quick overview of Altasciences' history. Founded in 1992, Altasciences has had a pretty large evolution over the last 10 years. When Chris Perkin was appointed as our CEO in 2010, we have put together a significant growth plan for Altasciences. From what you can see in 2013 and onward, is that there have been a number of acquisitions that have allowed us to add significantly to Altasciences' offering and the services that we can share with our sponsors. Our clinic in Kansas City was added in 2013, a large preclinical safety testing facility in Everett, Washington in 2018, and the acquisition of our CDMO site in Harleysville, Pennsylvania in 2020. You can see how we have purposely thought about how our services need to be intertwined and communicating with one another to deliver the best service to our clients. It all begins at discovery. We offer full bioanalytical testing capabilities from non-GLP to GLP. We have the ability to cover all areas from development into post-marketing studies. The majority of our work happens in the early phase, so we see preclinical from lead candidate selection and continuing support for your program into phase 3. The clinical work we do starts at phase 1, generally ends at phase 2 or 2A proof of concept studies. We also provide the full range of NDA-enabling studies and post-marketing requirements for our sponsors. In terms of research services, we support all operational activities with a very strong team of project managers, PK scientists, biostats data management, and medical writing teams that can help you from writing your initial documents, reports, and filing for market authorization. Our manufacturing and analytical services can take you from proof of concept, help guide you with what would be required to get you up and going from a drug perspective, and then they also carry you into commercialization. These are the services Altasciences currently offers.
Thanks, Angel, for that great introduction. Now let's talk about the pharmacy operations. Our integrated pharmacy services in Montreal, Kansas, and LA include highly trained pharmacists with multiple years of experience in clinical trials, from providing input on protocol writing, pharmacy manual development, dispensing, compounding, and aseptic techniques. Our state-of-the-art facilities are equipped to permit us to successfully execute all types of pharmacy preparations, from basic solid oral doses to sterile and non-sterile compounding. Our pharmacy teams have prepared all types of IP with different routes of administration, like topical, ADF studies, ocular, and inhalation. So whatever route of administration you're using to administer your product, our experienced staff have done it multiple times in the past. Our pharmacy activities follow GMP principles, GCP, and ICH guidelines. Compounding will follow USP 795 for non-sterile, USP 797 for sterile preparations, not to mention local FDA and Health Canada regulations. Our facilities are equipped with negative pressure HEPA-filtered ISO 7 clean rooms, ISO 5 biosafety, and laminar flow air cabinets. We are well versed in the regulations surrounding the import and export of investigational drugs of all types, and with the support of our logistics teams and brokers, will ensure timely customs release and shipment of your IP. Our Montreal site is licensed for controlled substances, precursors, and cannabis drugs. We regularly submit for controlled substance import licenses and E56 exemptions for studies involving scheduled drugs. For these types of studies, when the IP is shipped within Canada, we can also support QPIC to QPIC drug transfers. In the US, all controlled substance procurement and management are done through DEA licensed physicians Schedule 1 licenses. They also are licensed for Schedule 2 through 5. Our processes were submitted through rigorous DEA inspection. Our US sites saw DEA agents coming in and ensuring that we are up to code. Even though the US and Canadian sites have different regulations, we still maintain harmonized SOPs to ensure that all sites have a comparable work process and that we are able to provide anything that the sponsored study will require, no matter the local laws. If there's a limiting factor to conduct the study within one country, one of our other sites can swoop in and save the day. Having multiple sites with the same expertise and ways of working gives us a lot of flexibility. This gives us more options to find solutions to our clients' needs to successfully conduct their studies. So as you can see, all our sites are equipped to handle complex processes and procedures that are required for the study protocol. For example, our Kansas site moved into new installations in 2016. The pharmacy was designed with all the complex Phase 1 preparation needs in mind. The pharmacy teams are often integrated early in setting up a new study. When a potential client reaches out to our business development colleagues and have needs that are related to the IP or API preparations, our pharmacists are contacted and discussions with the potential clients can be had even before the contract has been signed. We do this to ensure that we start off on good footing to avoid delays due to stability issues or other. Beyond this step, the pharmacy and the rest of the clinic will provide feedback on the protocol to flag any potential risk we can see based on our past experiences before it sends to the IRB or the regulatory body. We follow very tight timelines to get your study done on time, so getting the IND or NOL within the required window is very important to us. As previously discussed, we can support import and export of IP in and out of the US or Canada. Our sites can also source comparator drugs, RLDs, or other products and materials required for study conduct. If a Canadian trial requires a US RLD, for example, we can ensure import from one of our US sites using a controlled shipper. Our labeling SOPs cover the different requirements for FDA US studies versus Health Canada ones. For example, our Canadian site requires labels to be in English and French. So our Canadian colleagues are available to review the sponsors' labels and provide the French translation in order to meet these requirements. Our sites, especially the US ones, have extensive experience conducting FDA studies. We conduct many EMA studies, especially in Canada, and are familiar with URDRELEX Annex 13 requirements. We also work with TPD and Visa, MHRA, regulatory bodies, just to name a few. Our staff are knowledgeable on the different requirements for each, whether it's retention samples, labeling, GMP requirements, and other things like that. Our sites are expert at developing pharmacy manuals and can help the sponsor develop the optimal way to safely prepare their IP or API. We can develop a draft version and send it to the sponsor for review. Multiple back and forth and meetings can be needed to get the final version of the pharmacy manual. If the sponsor already has a pharmacy manual, our team really likes the opportunity to review and comment on them to ensure that they are optimal and work with our processes. In certain cases, we can do mock runs to help develop the procedure and send samples for analysis when this cannot be done by the sponsor. A plus to having harmonized SOPs is to permit our different pharmacy teams to work together and problem solve. For example, when we have a client with multiple studies across our 3 sites, our processes are so similar that when reaching out to our colleagues in the other pharmacy teams, we can hit the ground running and find solutions to any problems fast. This also provides us with a larger pool of pharmacists' expertise and helps for brainstorming solutions. We also have rigorous QA and QC departments that are used to handling audit inspections from various regulatory bodies. We are often audited by FDA, Health Canada, EMA, and many others. Now I'll go over some case studies to show the complexities of some of our trials and illustrate the way we can support our clients in their preparation development. So for the first case study, we had a sponsor who came to our Montreal site with a novel API powder for oral administration that didn't really have a finalized formulation yet. To put you in perspective, this was for a phase 1 first-in-human study. It was double-blinded, placebo-controlled, with an adaptive SAD-MAD cohort format, and also included sentinel dosing. In this case, the Montreal pharmacy team had to closely work with the sponsor. A representative from the sponsor came to our facilities, and together we developed a vehicle to suspend the API. Our pharmacists were the ones that put together the pharmacy manual compounding procedures based on the mock preparations that were conducted in association with the sponsor. And then it was further used for dispensing during the study conduct. Multiple mock preparations were required at multiple dose levels, from the lowest dose to the highest dose, for the different formulations that were also sent for analysis. These analyses were to ensure the concentration of API were within acceptable limits, that there was no microbiological activity in the preparations. These tests also provided us with stability of the formulations that were included in the pharmacy manual. Since this was a blinded study, we needed to also develop a placebo formulation that not only looked like the test formulation, but also had a similar taste to avoid potentially unblinding subjects. At first, what the sponsor had proposed was to use a commercial product that didn't really mimic the characteristics that we saw with the API suspension. So we tried to find an inert comestible product that precipitated and looked like the API powder within the vehicle that we had developed, which is what we ended up using for the trial. It was pretty close visually. It was a pretty good match, except it precipitated the placebo maybe a little bit faster than the API. So to ensure that that was not visible to the dosing staff or the subjects, the prepared doses were put in amber syringes. These syringes were put in amber bags that you could not see through. And before administration, the clinic staff was instructed to resuspend these syringes. And the study was conducted and the blind was never broken because of this. Also, the API had a bitter taste. So we needed to ensure that no unblinding occurred if the subjects discussed the taste amongst themselves. So to resolve this issue, we added sucralose sweetener to each formulation. In the end, the bitterness was no longer noticeable. In this case, we also had Health Canada that requested a copy of the pharmacy manual prior to authorizing the execution of the study. So thanks to our pharmacists' hard work in collaboration with the sponsor, the pharmacy team helped get this study approved by Health Canada, and we're able to proceed with study conduct. It's not common for the pharmacy manual to be required for CTA submission to get a study done in Canada, but it's something that we're seeing more and more in the past few years. The second case study is an example of how our sites are able to work together. A few years ago, we had a human abuse study that involved a European client. They reached out to us to see if we were able to conduct a nasal insufflation study for one of their controlled substances products. At that time, the Montreal site hadn't had any previous experience with abuse deterrent formulations called ADF. So the clinical team at our Kansas site were brought in the loop early on and were able to provide their experience to support the Montreal activities for the ADF trial. The dose preparation was very complex, requiring extensive cutting of the IP, use of a sieve, retrieval, and weighing of the IP powder. Each dose could take up to 1 hour to prepare, and one dose, I mean one tablet being crushed, could take up to 1 hour. So these extensive manipulations that were required to ensure that the particle sizes were within the range that were needed, that the total weight amount of powder insufflated respected the method that was used. So because of the complexity, we had to train multiple pharmacists of both sites to be able to have a flexibility and cover all of the study's needs. The Kansas Director of Pharmacy was also present at the Montreal Pharmacist Intensive Training with the sponsor. SAS pharmacists needed to pass the sponsor-approved testing to be able to prepare the IP with the approved ADF method. So to conclude, this was a good example of the strength of the Altasciences network. With harmonized SOPs and procedures and experienced staff across the board, we can provide flexibility, efficiency, and consistency in the execution of clinical trials. At this time, I'd like to turn the discussion over to Angel.
Thank you, Kevin, for giving us the overview on the pharmacy operations. So we will talk a little bit more about the CDMO service and manufacturing and analytical testing that we have to offer. The Altasciences CDMO facility is located just northeast of Philadelphia. We have a 64,000 square foot facility that is composed of 2 buildings on 1 campus. Our main goal is to simplify contract services for our clients. We are able to provide our clients a proof of concept, research and development, clinical, and commercial manufacturing. We also offer different dosage forms to suit our clients' needs. A general overview of our facility: we have 15,000 square feet of warehouse divided between the 2 buildings, 3,500 square feet of grade D GMP manufacturing space. We have 2 additional GMP manufacturing areas that are approximately 6,000 square feet combined that are grade C areas. These areas are also capable of handling potent compounds. We have 1,500 square feet of R&D formulation lab and 8,000 square feet of analytical laboratories. We also have the ability to do expansions on campus for additional warehouse, manufacturing, or analytical spaces. We like to plan for success very early on in the project. With a request for proposal or a scope of work, we want to ensure that all necessary steps are addressed at the beginning of the program. If a scope is not identified, we will have several meetings with the potential client to give them insight and guidance on what is needed for each phase. We like to lay out the scope of work in a good flow from beginning to end. This starts with raw materials coming in and determining what type of method work is required, whether it be developed from scratch or transferred in. For manufacturing, we would discuss feasibility batches, engineering batches, scale-up batches if they are needed, and then clinical supplies. Quality assurance is also a critical aspect that is built into these processes. We want to have an easy-to-follow roadmap for our clients at kickoff. The kickoff meeting will finalize the timeline of the project that we will have regularly scheduled meetings to provide updates and feedback on. With our roadmap, we also like to enforce the fact that there is flexibility. As items come up or scope shifts, we want to ensure that changes can be made as a partner team and does not create any significant delays. One of the key product optimizations that we offer are liquid-filled capsules in either gelatin or HPMC capsules. We have the ability to create semi-solid hot melt products or liquids to be filled. A benefit of this is to allow first-in-human capability very quickly with minimal development efforts. In order to do this, we can melt down some high molecular pegs and suspend your API to get into the first-in-human trials. There are also some very interesting, unique excipients that we can use to improve solubility and target oral solid dose. The ability to help with abuse deterrent potential can be achieved with other targeted excipients. Nanomilling is another optimization niche process that we specialize in here at Alta. This process can also lend itself for bioavailability issues, which you can see with many APIs. This just provides another option for clients to get their drug into another dosage form that can help accelerate their timeline into clinic. Nanomilling allows us the ability to mill the API down to 20 nanometers. With our formulations lab, we have the ability to nanomill API in much smaller batch sizes to conserve API prior to any scale-up activities for proof of concept that will mimic the more complex equipment that will be used within the GMP manufacturing areas. From the R&D to clinical batches, the client has time to evaluate if any patents could be necessary or applied to their product. Another niche area that we offer and optimize is potent handling. We have a separate facility with the appropriate systems, airflow, and engineering controls for employee exposure that allow us to handle potent manufacturing. We do have isolators if needed. However, we try to design the process around closed systems if the process is capable of being completely contained. We will evaluate all compounds on a case-by-case basis for the optimal way of handling their product. Our potent area also doubles as our grade C GMP manufacturing area. We manufacture terminally sterilized products in this area, typically in the form of IVs. This type of manufacturing is preferred by the FDA if your product can withstand sterilization. The sterilization will help ensure that the product is suitable for injections. This is also a significant cost savings for our clients, as terminally sterilized products can be approximately half the cost of an aseptically processed product. Throughout the timeline of the product, we ensure that a Gantt chart is followed that includes the clinical team for transparency. The project management team will track deliverables and update timelines with the client input throughout the lifecycle of the project. API is one of the main deliverables that need to be monitored, as it can be one of the main contributing factors to impact the timeline of a project. This could be method or manufacturing delays from lack of API supply. The next piece that we monitor as far as the Gantt chart would be the method transfer, qualification, or validation within the analytical lab. We always evaluate the compatibility of our equipment. If the methods are qualified somewhere else, we want to check for like equipment that we transfer the method in. If the equipment is not like for like, it's not an issue, but we would need to qualify methods instead of performing a transfer. We also like to look at the robustness of the work that has been performed prior to Alta. The more robust the program, the greater chance of success for our first shot to ensure the timeline is not at risk. As the project progresses, the method would need to go from qualification to validation. There is the possibility that further development would need to be performed, as the requirements for phase 1 or phase 2 could be made more robust for phase 3 or commercial to complete the validation. In the manufacturing spaces, the robustness centers around the capabilities from our equipment standpoint. We like to ensure the equipment is here before the program starts, but if not, we identify the equipment that is needed and we'll get that equipment. As part of the quote, the equipment will be built in, and this allows us to go from phase 1 small batches through scale up and commercial. We do have various types of equipment from liquid fillers, powder blending, nano mills, and others to complete manufacturing campaigns. We like to keep the time to get into manufacturing in a very realistic timeline that meets the client's needs. Having projects way out in the future does not help a client achieve their goal of time in the clinic. And one of our case studies around kind of our speed that we offer here at Alta is we had a client with an extremely aggressive timeline and a challenging situation. We were first approached in August, the first week of August, with a request to have product in the clinic by the first week of October. The client came to us with an API and no formulation development work. All that was known was that it needed to be nano-sized and the time to get into clinic. We had around 8 weeks to go from scratch to a first-in-human trial. We had our kickoff meeting, and we were off to the races. We developed 5 formulations and put them all on accelerated stability. While the formulation was in development and accelerated stability phase, the lab had a sample and began working on method development for assay and impurities. After the 2-week accelerated stability, we took a look at all the formulations and decided to continue with 3 formulations for 2 more weeks on accelerated stability. Once that was completed, we selected 1 formulation as the lead candidate and finished the method development work for the product. Protocols were drafted, reviewed, and ready to be executed to prepare for GMP supplies. Within the accelerated timeline, the client also wanted us to produce this product down the road for future proof of concept. So we needed to make sure that the formulation was robust, because that was a key and essential feature. As we scaled from the formulation development work, we did have to source 1 piece of equipment. We were able to locate, source, qualify via design, installation, operational, and performance qualifications in-house. The batch was ready to be made at the beginning of September and went all the way through labeling. We shipped the material after release to the clinic sites across the country. The client was able to get their product as scheduled and into the clinic, which made them extremely happy. This was viable due to the client input and responsiveness, being they were on site during testing and manufacturing in case any issues arose. We were able to rectify those quickly. The aggressive timeline was definitely stressful, but we want to do what we can in order to meet our clients' needs. The first human trials went very well, and over the course of the following 6 months, we were able to manufacture their clinical supplies. So we're going to jump into the Q&A portion of this webinar. So we have some questions that are going to the chat box, and the first question that we have is, how do you handle dose changes or potential formulations within a trial? Kevin?
So there are 3 points that we can focus on. The first being with the communication. We need to have good communication between ourselves, the sponsor, and our CDMO group. That's the nice thing when we all work within Altasciences. Just a simple email or phone call with our CDMO team in Philly, and we can quickly get a plan together. Then we can present that to the client, and we can move forward with the next steps. Once that initial communication has been established and we have our plan, we can then proceed with our batch method of development. When that's complete, of course, we can go through sponsor approval, IRB approval, and all the other regulatory agencies that are involved. And lastly, we'll need to create samples, whether it be oral suspension, sterile products, solid oral form. Those samples will be sent for analysis. So whether it be an outside vendor or back to the sponsor, those samples need to be tested for concentration, endotoxins, sterility, and other required validation methods to be performed prior to dosing formulation. That's all to make sure that we're within the limits of the required batch method that was developed.
For the CDMO, we have the flexibility of working in our formulation development space or GMP suites. The formulation development allows us to quickly perform any small-scale studies without the need for cleaning methods and large quantities of materials. Within our GMP suites, the cleaning method gives us the flexibility to incorporate different pieces of equipment to ensure that the optimal formulation is obtained. Understanding the maximum and minimum doses helps us ensure that our cleaning limits are appropriate and that we will utilize appropriate controls for the safety of our team. Once the doses are made, the analytical and quality teams can be in a good position to get the product released quickly and back to the clinic.
Okay, let me read the next question. When should we engage the Altasciences team for early planning and engagement?
For the CDMO side, the earlier we can be brought in, the better. It allows us to have a full understanding of the program as well as time for our relationship to mature. We can discuss all documentation requirements and deliverables early on so that we understand what is needed for changes along the life cycle of the product. Every time you work with a new client, you have to get a feel for how things work and how they need to be presented to one another for ease of review. The more time we get to spend together enables us to get through any rush or urgent situations with fewer issues.
For the pharmacy side, while I agree that the earlier is the better, we want to do things the right way and make sure that the sponsor meets all his milestones. So it has to be done in a way that ensures the safety of the subjects and the integrity of the study. So speed is something that at Altasciences we kind of pride ourselves on, but it's also the quality of the work that we do. So the earlier we get engaged, the better. And that's a nice thing when we're working with our CDMO group. We can really coordinate and we're used to efficiently working together. So we're able to find solutions and expedite the process and procedures needed depending on whatever protocol we're going to work on.
Next question just popped in. Can you source comparator drugs?
So that would be for us. So yes, we can. It's a service that we provide regularly and often to our clients. We source reference drugs normally from our approved local licensed pharmacies. So if it's a RLD from the US, we'll source it from our sites in the US. If it's a comparator, it can be sourced also from Canada. In some cases, reference drugs are not always available at our regular vendors. So we can also get into discussions, let's say, with local manufacturers to source a drug. I have in mind a recent DDI study that the product was sold in Canada, but only through specialty pharmacy services. So we were, the sponsor and the manufacturer were in communication with us, and we were able to get that product to be sold to us, even though it didn't respect the specialty pharmacy's way of normally selling the product, and we were able to purchase it for the study that we conducted. So for the next question, depending on the protocol, we have different blinding solutions that can be provided. For example, we can use a matching placebo. When this isn't an option, we can over-encapsulate the IP or active control when conducting double-blind studies. Placebo would simply be the same capsule, but filled with the inert filler instead of the tablet that we're over-encapsulating. This way, while the clinic staff and subjects cannot identify the treatment being given. Depending on the administration route, we can also use oral amber dosing syringes, which are used only for the oral solutions and suspensions. If it's for subcutaneous IM or IV, well then the syringes are clear, so we have to add a amber shield to mask the coloration, and we can add amber bags over the extension sets and the IV bags, also to hide the different colors of the placebo versus the active product. Sometimes for completely different IP administration routes, we'll need to have a double dummy method. So all different possibilities depending on the type of study.
Another question just popped up. Can your sites perform over-encapsulation?
I quickly just touched on that. So yes, that's something that we do for blinded studies. We have a specific SOP that covers over-encapsulation of tablets. For other types of over-encapsulating, let's say we're over-encapsulating just a single powder in a capsule, we can write a pharmacy manual that will cover all of these steps needed for the over-encapsulation. So this would require that over-encapsulating API powder needs to be weighed individually into each capsule. So yes, we can do all sorts of over-encapsulation of different types of study protocols.
In the CDMO, we can also perform over-encapsulation within our GMP suites. We're able to perform this with or without a backfill, just depending on whatever the client needs.
I'll go for the next question. It's about scheduled products. It's asking if Altasciences can handle Schedule 1 products and also cannabis. So maybe I'll start off. So for all of our 3 clinical sites, Montreal, Kansas, and LA, are all able to handle Schedule 1 products as well as cannabis. We have extensive experience with controlled substances, precursors like pseudoephedrine, and cannabis. We've done lots of studies with cannabis of all types of administration. We've done vaping studies, oral oils, capsules, tablets, name it. We've pretty much done them all. So yes, we're used to working with all these controlled substances, including cannabis.
And we are also able to handle schedules 1 through 5 here for the CDMO as well as cannabis. We have a couple clients in that space right now, and as legislations change, we continue to stay up to date with all the requirements and the DEA controls as well. Looks like the next question just popped in. Do we have ways to develop and manipulate abuse deterrent products?
Okay, so for these types of preparations, it's really very specialized. We have to do extensive manipulations of the controlled substance in a way to simulate the way that a drug addict would prepare their doses, but it also has to be in a way that is standardized and replicable for the study. So when a sponsor doesn't have a validated method to manipulate their product, we need to work closely with a specialized vendor to ensure that the method meets the specifications that are set forth by the FDA guidance on abuse deterrent formulations for solid oral dose forms, and also specific requirements for opioids. We also worked in the past with sponsors that have their own validated method without passing by the vendor. Either way, we want to really ensure that the IP samples are within the specified range before starting the clinical trial. So it's very important that our staff has proper training on the specific method, that we prepare test samples that will need to be analyzed to make sure that they meet the specifications outlined in the guidance. So it's very pharmacy intensive studies that are quite interesting to do. Do you want me to read the next one? So I see, how do you anticipate and handle products to eliminate cross-contamination?
Here for the CDMO, our cross-contamination mitigations really are focus anytime we're within our GMP suites. Cleaning methods get put into place prior to taking any product back into our GMP area, ensures that we're able to clean anything that it does touch, and eliminating and avoiding that risk. We also have HEPA filtered air within our suites with appropriate airflows, preventing any aerosolization or contamination from escaping into any of our adjoining rooms.
On our side, we are not GMP certified, but we do follow GMP principles. So cross-contamination is something we work really hard to avoid by following GMP principles in the way all of our products are manipulated in our pharmacy. We really segregate the different products. Our SOPs clearly detail the line clearance steps to be performed before any pharmacy activity. Only one product can be kept in one working area at a time. Areas cleaned after each product is manipulated. And for example, for oral dose, new pill counters, we'll change our gloves, we'll clean the area between each product that's dispensed. And if there's a placebo, we'll start with the placebo, since there's no active ingredient, and we'll finish with the active ingredient in that dispensing activity. All IP dispensings and preparations are overseen by a qualified person, usually a pharmacist. And if any processes are not respected during the dispensing activity or compounding activity, well, the IPs will be quarantined and not provided to the clinic, and we'll start again a new dispensing activity for that dosing. Next question, do you go beyond early development or commercialization?
Yep. So really our focus here is we want to be an end-to-end or a one-stop shop for our clients. So getting integrated with Altasciences at the CRO stage, starting your clinical trials, moving everything you need to do there, and then having the kind of handoff for when your batch begins to progress to the CDMO space. It really allows us to have streamlined communication with an overarching project manager. So basically you'll have one point of contact that will be able to kind of manage your program, whether it's in Montreal, Philadelphia, Los Angeles, or Kansas. and it really helps kind of focus our efforts on what we're going to do and ensure that the deliverables are being met. We want to be able to handle all stages of the formulation, development, clinical, and then commercialization for our clients. So yes, we do go from early development to commercialization and really trying to use our network from end to end and really streamline your process and try to get your product to market.
Okay, the last question that we have seems to be for you. So it's a general question, comment. How do you streamline the process in early development?
So when we're looking at early development, we are working with some of the smaller startup companies, and really commercialization is the furthest thing on their minds. However, when we are doing our project kickoff and throughout our client meetings, we really want to give some guidelines as far as what's needed in the future to kind of avoid having to do any rework and save some cost. We want to focus on the formulations and ensure that they're within a good design space for robustness. So this allows us to understand the size and the intended target population. We can do some scaling based off of which phase of the life cycle it's in, phase 1, phase 2, and then as the project begins to grow, we've already done some of the legwork up front, so we're not having to go back, restart stability studies or anything of that nature, and really just kind of continue it and watch it grow throughout the life cycle. And it looks like I think that's all the questions that we currently have. So we want to thank everyone for attending the webinar and submitting the great questions. And thank you, Kevin, for doing this with me and taking some time. I know you got a very busy schedule, so I do appreciate it.
Same to you. Thank you very much, everybody.
Thank you all. Have a good day.